Welcome to Biol102, my dear students. And today I'm going to talk about the mitosis and the cell cycle. Now you know that some of these organisms are only made up of one cell. They are called unicellular. Most of the organisms you are familiar with are made up of many cells. They are called multicellular. They are called multicellular. Okay, so here how they become multicellular from unicellular. This is what you are going to learn today, and this is possible because of cell division. Now, before we talk about the cell division, I want to talk about the cell cycle. And here in this PowerPoint, you can see this cell cycle. This cell cycle is divided into interphase, which takes up more than 90% of this time of cell division. And then next is mitosis. Mitosis is the division of nucleus. And then there is cytokinesis. Cytokinesis is the division of cytoplasm. So the cell division can be divided into interphase, which takes most of the time, mitosis and cytokinesis. Let us talk about them one by one. Interphase is sometimes called resting phase, but actually the cell is not resting. There are a number of things that are going on in interphase. So let us first learn about the interphase. So here, whatever I have said is explained in this cycle, that when the cell divides into an orderly series of cycles known as cell cycle. This definition of cell cycle. Cell cycle is divided into interphase and mitosis is divided into four phases. Mitosis is divided into four phases. Usually in plants, this mitosis is studied in onion. So we are going to study this mitosis in onion cells. In diagrams that you see in some of the textbook on zoology, they describe mitosis in animal cell, but here we are going to describe mitosis in a plant cell, and that is onion. Okay, as I have said, interphase may occupy 90% or more of time. And usually it is hard to find these stages of mitosis under the microscope. I will explain this to you in the lab. So here is again is the cell cycle. You can see the interphase, mitosis, and cytokinesis. Interphase is divided into G phase, S phase, and G2. So I'm going to describe this G, S, and G2. And in mitosis, I will describe prophase, metaphase, anaphase, and the telophase. So here is interphase. So we say that when the cell is not dividing, they are in interphase. And for many years, it was called resting. And then he asked this question, are they resting? The answer is no, they are not resting. There are a number of things that are going on in the cell, but we cannot see them. So there are three consecutive periods of intense activity which takes place during interphase. And these three are gap, growth, G1 phase, synthesis, S phase, and this G2, which is again the growth phase. So G1, S, and G2 are the various phases of the interphase. What happens in G1 a period is that it is relatively lengthy, and in this period the cell will increase in size, ribosome, and substances that either inhibit or stimulate the S period are produced. During S period, the unique process of DNA replication or duplication takes place. This is very important because without this duplication or replication of DNA, division of cell, division of nucleus is not possible. In G2 period, mitochondria and other organelles divide, and microtubule and other substances directly involved in mitosis are produced. Coiling and condensation of chromosomes also takes place. So this is the end of interphase, which takes up 90% of the time. And most of the things happen in this interphase. It is not resting. There's a lot of activity during interphase. Now mitosis. Mitosis is the division of nucleus. And as I have said, all organisms begin life as a single cell. Question he's asking, which one? This cell is zygote. Every organism starts its life from this zygote. Mitosis is the division of nucleus, Cytokinosis, the division of cytoplasm. It ensures that cell divide and each cell get equal amount of DNA. It is quantitatively and qualitatively equal division of the hereditary material. In higher plants, mitosis occur in meristems. Where are meristems located? Meristems are located at the tip of root and at the tip of shoot. In the class, I will explain to you with the help of diagram. This is what you are going to study next. Mitosis takes about 5 minutes to 3 hours, and as I have said, there are 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. Let us start with prophase. Prophase is the longest phase of mitosis. In this phase, chromosome becomes shorter and thicker and two-stranded, and this double nature becomes apparent because of duplication of this DNA during S subphase of interphase. Nuclear membrane and nucleolus disintegrate and they disappear. Chromosome becomes shorter and thicker. So duplication can be seen as I have said. Each chromosome is made up of 2 strands or 2 chromatids. The centromere, which is the dense constricted portion of a chromosome to which a spindle fiber is attached, holds each pair of the chromatid. The centromere becomes double, as I said earlier, in G2 phase of interphase, a kinetochore. Now this term is important. Kinetochore is a dense region composed of a protein complex which is located on the outer surface of each centromere. Spindle fibers become attached to the kinetochore. Chromosome may have other knobs like constriction and are called satellites. Nucleolus now becomes less distinct and eventually it will disintegrate. Eventually the nucleolus will disintegrate. Nuclear membrane disintegrate. By the end of prophase, the spindle fibers develop. In animal cells, centrioles are present and they are outside the nucleus. And they play a role in spindle formation. Centrioles have not been detected in plant cell. In animal cells diagram, you can see these centrioles. Metaphase. This is the main feature, and in this case, there is an alignment of this chromosome at the center of the spindle, which is called equator. Then the next phase is anaphase. It is the briefest phase of the phases, and it involves the sister chromatids of each chromosome separating and appearing to be pulled to the opposite poles. Daughter chromosomes move toward the pole as their spindle fibers gradually shorten. Centromere of chromosome assume V shape and all chromosomes separate and move toward the pole. I will explain this to you by making diagrams on the whiteboard, and then when you will make these diagrams, this will become more clear to you. The last phase is of mitosis is telophase. There are 5 main features of telophase. Each group of daughter chromosomes becomes surrounded by a reformed nuclear envelope. The daughter chromosomes become longer and thinner, and finally they become indistinguishable. The nucleoli reappears, the spindle fiber disintegrate, and the cell plate is formed. The set of fibers appear at the center of the plate and they form phragmoplast. This term is also important, phragmoplast, set of fibers at the center of the plate. Then division of this cytoplasm takes place, and this is what is called cytokinesis. I told you when I was talking about the cell wall that there is formation of this first partition, that is middle lamella, and this happened in this cytokinesis. Here next diagram tell you about this chromosome. In the beginning, it is made up of only 1 chromatid. But during the S subphase, it is duplicated or replicated. So you can see 2 chromatids which are attached to 1 centromere. These are called sister chromatids. Here is again a chromosome, how a chromosome look like. There is centromere, and attached to this centromere are these chromatids. And here you can see the 2 chromatids attached to the centromere. They are the sister chromatid. This is a diagram that shows cytokinesis, the formation of cell plate. So you can see the partition in the center. Later on, it will develop into middle lamella, and after this, there will be formation of primary wall. So this is the end of this slides. I will ask you to make diagrams yourself. I will give you instructions in the lab. Each student is asked to make good diagrams of different phases of mitosis: prophase, metaphase, anaphase, and telophase, with four chromosomes. Every student will show four chromosomes in the nucleus in the beginning, and then at the end will show that this cell has divided, and each cell now again has four chromosomes. Mitosis is the division of nucleus. One nucleus will divide into two nuclei, or one cell will divide into two, and these two cells are qualitatively and quantitatively equal. They have the same number of chromosomes, they have the same type of chromosome. So with this, we come to the end of this recording. This you will understand this by making these diagrams, and if there are questions, please ask this